Good afternoon, everybody. This is going to be a kind of a tale of a set of reactions based on this DCC system. Okay. So a little bit of a preamble or a prologue to this story. There are various kinds of hypotheses as to how prebiotic life could have emerged into the last universal common ancestor. But assuming that there was a hypothetical RNA world, the question remains as to how the hypothetical RNA world got to the LUCA stage. If we look at life from a top-down approach, one eventually goes down to the ribosome, which I strongly believe, based on the evidence, it's the largest white elephant in the room of prebiotic life and also the LUCA. If you look at the ribosome, you go deep down into the ribosome, you end up with a PTC, but it's almost like at the end of the ladder, when you look down, what do you see? What you see is a prebiotic world. Assuming that there was a hypothetical RNA world, how did we get from the hypothetical RNA world to the LUCA stage? So the current hypothesis is that the hypothetical RNA world was probably terminated by a proto-ribosome world, an ancient peptide-making machine. However, there's a small problem, the absence of a demonstrable replicase. Therefore, in the absence of this replicase, we have heard a lot about wet-dry cycles. In a similar fashion, we have this alternate system which was developed in our lab. It's called the dynamic combinatorial chemistry system, which uses cycles of synthesis and degradation, not much different from the wet-dry cycles. There's one big caveat, though. The enzymes that we use are not particularly very prebiotic, not at all. However, that's not the point. Whether we use enzymes or whether it was any other system of degradation and ligation, the RNA sequence acceleration pathways are likely to have been along similar lines. That's the central idea. Now, what did we do? We used two enzymes. One of them is the nuclease, a benzonase nuclease, which degrades all kinds of nucleic acids while having absolutely zero proteolytic activity. And when it does the degradation, it creates a 5' phosphate N and 3' OHN in the degraded product, which is perfect as a substrate for the following enzyme, which is a T4 RNA ligase 1, which catalyzes the ligation of these two products with the help of ATP. So what do? We put them together in a buffer system, which we establish, along with an RNA oligo. And while one enzyme cuts, the other enzyme puts it together. And this cycle goes on over a period of time. And the going assumption is that this will help in increase in buildup of the RNA size, if not complexity. I'm very nervous about using the word complexity after today's morning session. Yeah. So this is exactly what happens, shuffling of cards, but with one small difference. Assuming that instead of just one deck of cards, there are several decks of cards. Each deck of cards refers to one oligo in the entire mix. So every time you shuffle them, there is a new combination, and hopefully buildup of size, not complexity. We do not know yet. Okay, so what we did, we chose the seed oligo, which is a reasonably humble oligo. It has no secondary structure, so convenient, and it has just one small little repeat of A followed by 3 Cs, but absolutely no secondary structure. So you take the seed oligo, combine it with the benzonase and ligase along with ATP, run it for 180 minutes, take out samples every 30 minutes, sequence the same, and also run gels of each of these samples together. And what do we see? When we run the gel, we immediately see a 40-mer, which we suspect to be the oligo ligated with itself. And we also see bands that gradually increase in intensity, but past the 120-minute point, they start decreasing because the ATP gets used up. Okay, when we sequence these reaction pools, we make DNA libraries, which had its own set of problems. I'll not be talking about them. But when we look at the sequences, the blue bar here is the seed oligo, which decreases over time, which one would think is intuitively expected. But what is interesting is the increase in the number of unique sequences, which is unique to each time point. At 30-minute time point, there is a set of unique sequences that are compared to the seed oligo, they're at this level, and then they keep on increasing in percentage. This shows the percentage. And at the 120 and 150 minute points, they are almost more than 85% of the total RNA pool. But as we saw in the gel, past the 120, 150 minute point, ATP gets used up, so the total overall RNA pool starts getting degraded because the benzene takes over. It's almost like a catch me if you can experiment. So what did we learn from this particular experiment is that the DCC system definitely produces products larger than the seed, and point mutations are very, very rare in this system, and most of the changes involve insertion and deletion of small blocks akin to recombination events. However, if only we could manage to figure out a way to keep on adding ATP, we'll have more room for extra RNA sequence space exploration past the 180 minutes. Okay, now it is not realistic that in the ancient RNA world, if there was one, that there'll just be one oligo. What if there are multiple oligos? So we use multiple seeds. What we chose was this mini helix alanine tRNA, which is, we just split it into 4 different parts and put them all together, hoping to see what happens. And what happened? After 15 minutes, distinctly, this particular section with the CCAN completely disappeared. You cannot see it anywhere here. But after 15 minutes, you still see the other 3 on top. But after 180 minutes, you find that these three have been ligated together, minus the 4th part, and this one was nowhere in the picture in the initial RNA pool. They have now occupied amongst the first top 10. Now, the original seed from which the 4 oligos were derived had just 1 copy number. That kind of reverts us back to the Stephen-Gouldian paradox, as I call it. If you were to start life with pieces of the ribosomal RNA with something like this, or even wet-dry cycled, it's highly unlikely you will get something like the pre-PTC. So that's a huge challenge we have to keep in mind. Okay. Now, it is also a reasonably fair assumption, based on all the talks we have listened to, that the RNA world was not just an RNA world. It must have had inputs of peptides or amino acids. So how do the presence of peptides or amino acids could have impacted this sequence-based exploration? To do that, we chose 2 peptides. One, a peptide of ancient origin, which is highly conserved extension of the ribosomal protein L2. And as a control, we chose a second peptide, which is of much more recent origin, which is FMLP, which plays an important role in the human immune system. It's a short peptide. And we did the same by putting each of these separately into this DCC system with the 2 enzymes along with the oligo. What do we see? We see that while L2 inhibits formation of the larger product, FMLP appears to favor the ligated product. We do not know why, but this is a very good sample to just show how the presence of peptides or amino acids could impact sequence-based exploration when without them. Now, looking ahead, we are thinking of using the DC system with RNAs as well as prebiotically relevant amino acids. These are amino acids which have been cataloged very well by Paul Higgs and as well as Irene Shen's group, and as well as using combination of these amino acids in short peptides. Again, I have to make this caveat that prebiotic systems did not have these enzymes. We are only interested in the dynamics of the RNA sequence-based exploration, right? Now, here is the epilogue. It's a seminal paper from Carl Woos. In an attempt to determine what sorts of prebiotic interactions between polynucleotides and polyamino acids or their derivatives are plausible. While doing so, we hope that this knowledge will in turn lead to the development of a concept of what a workable, primitive form of translation could have been like. So towards this visionary goal of the late Carl Woos, I think it is important for us to trace the roots of the ribosome through various systems, including the DCC. That's all I have. Thank you.
Are there any questions out in the audience? If anyone's gathering up the courage for it, I actually have a question. The sequences that you do see that are showing up, are they correlated with any particular property that causes them to show up? Because it seems like in terms of sequence space, There are
Okay.
so many possibilities. So is there any commonality for what keeps showing up?
There are
or
a
what?
few more slides that I removed because I might be running out of time, which is a very good habit of mine. So benzonase, the enzyme is not particularly agnostic. It's very, we found that from the statistics, it cuts at anywhere after or before a C. So that could be the reason why they are appearing. However, if we also tried a different system by using alkaline hydrolysis combined with another ligase, which combines 3' phosphate with 5' OH. But that enzyme, number one, it forms cyclical products. It doesn't push the reaction forward. Number two, it's ridiculously expensive.
All right. Dr. Tirumalai, thank you very much.